Okay, hi everybody. To start off, I am going to change the test date. I was going to have it on Wednesday the 20th, which is next class, well, after Friday. But when I said that and you all basically had a heart attack, I do not want to stress you out. My goal in this lab is that you understand it, you get what we did, why we were doing all of those steps, not rush to memorize something for a test, but to understand it. So, I'm going to move the test back. Unfortunately, well, fortunately and unfortunately, we have a day off next week and the Natural History Museum, so I only see you once, which is going to be on Wednesday the 20th, which is when it was going to be. So what I'm going to do is, on that Wednesday, I just took that other lesson, I'm going to switch them, and then we'll start off class with a little bit more Amgen review. That way you can ask questions during that day as well. But I'm going to make this video to help you study for the assessment, and so that way you are very clear on what you need to know from the lab. Now, most of the concepts that we just touched on we'll continue to talk about and learn about, so you don't need to go out and learn something new. What you're responsible for is what did we do in this lab, why did we do that, and then those basic concepts that were involved in the lab. So I'm going to just take you through my lab manual. That way I can remember every lab that we did and stay in order here. So to start off, all of our labs that were very relevant, even if they took maybe 2 class periods, they boil down to 3 main labs: Lab 2A, 4A, and 5A. All the other stuff was either practice, it was preparation, it was allowing it to grow, and learning about what we were doing. So I'm going to start off with 1 because it built the background knowledge for Labs 2, 4, and 5. So to start off, some things that you really need to understand for the assessment are the technology and the equipment that we use. So to begin, you need to understand how to use a micropipette. I hope that you know this by now, but I'm going to start with the P20. So on the P20, there's the top, we have our, backwards there, our screen. Please remember that the bottom, even though it's hard to see in the video, the bottom number is red. That red number symbolizes the decimal point. So this is not saying 100, this is actually saying 10 microliters is the unit. Oh, this top one is broken. And now we're at 9.4, go a little bit more, 8.2 microliters, and so on. You should know how to read one of these little screens on the assessment. We also have a P200. This one was used for larger amounts. Now this bottom number is no longer a decimal, it is the last number, so this is actually showing 100. So when we have 2 set, and this is review from that very first day, here we go, a P20 and a P200 right next to each other, we actually have, where are we, 10 microliters in the P20, but 100 microliters in the P200. Remember, for these, you never want to pipette over what is written on the top, so you never want to go above P20 for the P20 and 200 for the P200. Other than that, you don't really want to go below 1 or 2 on the P20 and 20 for the P200. Sorry, when I see them, they're backwards in the screen, so above or below 20 in the P200. Okay, so that is the first piece of equipment. We familiarized ourselves with that a lot in Lab 1A, making sure you know that conversion of how many microliters are in a milliliter, and that all of that can be found in your packet that you've been working through. This is a really good tool to use to review. Make sure you review that biotech experience, what is genetic engineering, and then here are all of your conversions on the top of this page. What I'm really worried, or not worried about, but make sure you understand how many microliters are in a milliliter. Okay, let's see here. Like I said, I'm going to kind of flip back and forth on some of these just to make sure I hit everything. So let's start with Lab 2A. So 2A was titled, How Do You Begin to Clone a Gene? So this was the beginning of our lab, and this was just making sure that we had the right, oh, come on, sorry, I'm pulling my lab up. making sure that we had the right stuff to work with. So some things that you should review are the difference between chromosomal DNA and plasmid DNA, understanding the structure of a plasmid, and why is a plasmid ideal for biotech engineering. What about that plasmid makes it so useful? Understanding the basic components of the plasmid, things like the oocyte, the promoter, the gene of interest. I'm going to show you my screen in just a minute, or my board. You should all have this copied down. Let me move over. You should have this copied down in your notebooks, but if you don't, oh goodness, get closer here. Hold it there for a second. Okay, what you need to know from Lab 2A was the different pieces of our PRR plasmid. That was our plasmid that we were focusing on. Our oocyte gives us the ability to replicate, or not us, the plasmid. The RSC acted as the activator. The activator controls that promoter, which allows for transcription to occur. Our gene of interest down there at the bottom was that RFP gene that we were cutting out with the PBAD as well. We used two restriction enzymes, the BAMH1 and the HIN3. And finally, we have the AMP-R, which gives the plasmid ampicillin resistance. So making sure we understand that plasmid, what it looks like, and what all those little pieces are. So what is the oocyte? What does it allow the plasmid to do? And why does that allow the plasmid to be such an important piece of biotech engineering? So let me go ahead and check here. Make sure I covered everything from 2A. So this was our restriction digest. Where is that? Sorry, I wish they wouldn't give us so many pages. So this was our flowchart. You should have access to the entire procedure if you want to review. But basically what we were doing was we had our R plus and our R minus tubes, and into those tubes we put in, let's see here, the restriction enzyme. Oh, that's right. We did the restriction enzyme in the R plus, and then we did the deionized water in the R minus to show, so that way we didn't get any cuts in the R minus tube, in the plasmid of the R minus tube. So basically, this was just our restriction enzyme that we put in to our plasmid. That's all. That was called the restriction digest. Now the reason we did this was because we needed to ensure that we had the correct genes in that plasmid. So our gene of interest was that RFP that I showed you at the bottom there. So we wanted to make sure that we had that in the plasmid before we went to the transformation process. So that's why we did the restriction digest. Remember, we let that incubate in the water bath for an hour, and then I froze the tubes. So all that we did, essentially, restriction enzyme into the tube, the R plus tube that of course had the plasmid already in it, and then the other one, the R minus, water into it, put them in the water bath, put them in the freezer. Then the next lab was where we actually interpreted that data from our restriction digest, making sure you understand what it means to do a restriction enzyme digest, which is essentially we put in the restriction enzyme and it allows us to cut the plasmid. From your pre-lab, making sure you understand like what are sticky ends. You already researched that. Let's see. I think I covered it. Understanding that when we use the restriction enzymes here, we use two different restriction enzymes. We use the BAMH1 and the HIN3. The reason we did that was because we did not want the plasmid to reform into a circle after the digest, because then it would have been no good. So that's why we use two different restriction enzymes, was because we get two different sticky ends, so they're not going to be able to reform into a plasmid. I think we're good here on lab 2. Let me double check. Understanding that in the R+ tube, after the restriction enzyme, we should get 2 segments, the gene of interest and the rest of the plasmid, whereas in the R-, we should have 1 segment because it was never cut. Okay, I think we're good there. So now, lab 4. Lab 4 was making sure you've got a recombinant plasmid. So now we are using another technique that we practiced at the beginning in lab 1, which is our gel electrophoresis, which is an awesome tool that we have to be able to identify if we have the correct size and charge of fragments. So remember when we did the gel electrophoresis with the dyes, it was able to separate into the different colors based off of their charge and molecular mass, or molecular weight, pardon me. So what we are going to do in lab 4 is we're going to take the results from that restriction digest and we're going to run them through gel electrophoresis. So before we did that, let me go to my flow chart here. We added the loading dye to all 3 tubes. We had R+, which had the plasmid, the restriction enzyme, hopefully cut plasmids. We had R-, which had the plasmid and water. And then we have now an M2, which is going to be our DNA ladder. We use a DNA ladder to give us something to compare to in the electrophoresis. So we added the loading dye to all 3 of those tubes, and then we just ran the gels. And then this is where, and let me make sure I still have those pictures up. If you go to, let me see, can I flip this camera? Okay, no I cannot. So if you go to our course in Schoology and then you go to Amgen, I have the Lab 4A gel electrophoresis perfect. They're still up there. You can check out our results again from Lab 4A. Now what we had predicted was going to happen was kind of this, let me see if I can draw it out real quick. Oh, perfect. So we all drew this in our notebooks. We had predicted that we didn't know what our ladder was going to look like yet because that was new, but we had predicted with R- that it was going to be one line, not very far down because it was one plasmid, so it's big, so it's not going to move as far. And then what we had predicted for R+ was we should have gotten a 4,000, I'm going to write that down, 4,495 base pairs, and then 807. So then what we predicted for our R+ was that the 4,000 wouldn't move as much as the 807 because the 807 is much lighter. Remember, in electrophoresis, the things that are going to move the farthest are going to be the things that are the most negative and the lightest because they move easier through the electrophoresis gel. The reason that this works with our plasmids is because DNA has an overall negative charge. I say this over and over, DNA, N is for negative, so DNA has a negative charge, and that's what allows that to move so well in electrophoresis. Also, we can separate then our different segments based off of weight. So the reason we do 4A is because we want to find these results. We want to make sure that what we had already used in lab 2 is the right stuff before we transform our bacteria. So this is what we were looking for. We were a little surprised though because our results ended up looking kind of like this. Actually, they looked almost perfectly like this because we did a great job, which is awesome. So on, I don't know what side that would be for you guys. On my right here, you have the M. M was the DNA ladder. The purpose of it was to act as a comparison. So we can see that it spreads out into its different groups based off of the different amount of base pairs that it has. Our R+ was exactly what we expected because we had the 4,495 and then we had the 807, pretty much exactly where we would want them, and we almost, I think actually everyone got great results with this. The one that was surprising to us was the R-, and that was because we got 3 bands as opposed to just that one that we had predicted. So then we talked about the reason why we got those. And this is when I showed you our little models. So we have 3 different forms that our plasmids can be in. And I think I actually have a really good image as well. Yeah. So the 3 forms affect how quickly the plasmid is going to move through the electrophoresis chamber. So if we look here, the one that moved the farthest, we'll start there, was the supercoiled. So that means that something about that supercoiled shape allowed it to move down the electrophoresis chamber so far. So if we look at that image, I'll put it up in just a second, and I hold this up, this is what that supercoiled plasmid would look like. So remember, plasmids usually have that circular shape, but if it's supercoiled, it's wound really tight together. Just like aerodynamics of a car, it works the same way. Because it's so aerodynamic, we can say, it's going to move through the gel better. Now, obviously, it's not aerodynamics working with it, but you get the idea. So this is a better image here of that supercoil. It's that top one. So because of its shape, it's going to move very quickly through the electrophoresis chamber. The next one we have is the NICT. Here's the image of NICT. There's the NICT one, the second one. All right. Okay, so then the NICT circle is going to be open, and we'll move through actually fairly quickly still, especially in comparison to our multimer. And then it will have that small little opening in part of it. So that would be our NICT. And then finally, the one that moved the slowest was our multimer, and that's because it has several copies of that plasmid because it's in the middle of replicating there. These are going to be different colors, but pretend that they were the same, and they would be connected like that. So you can see that that's going to move much slower than something like that initial supercoiled that I showed you from before. So that shape will also affect how it moves through the electrophoresis chambers. But the 2 most important things are going to be negativity or charge and weight. So we did lab 4. We put our gels under the transilluminators, what that was called, in order to observe them, and that's when we got to see the glow in the dark. And we observed our gels. I posted the pictures, and they looked pretty good. So we're like, all right, we are ready to move on to our next lab. Let me pull it up here. Which was Lab 5A, getting a recombinant plasmid in bacteria. So it's important because we were given the PRR plasmid, but that plasmid was actually recombinant plasmid, which means that we took the DNA from a different plasmid and actually put that gene into it. So we have actually great samples here. So we had 2 plasmids that we were starting with. One was PCAN-R, and then the other one was the PRA plasmid. We are actually taking the information from these 2 plasmids and then combining it by taking a gene from the PRR and the PCAN-R, the PRA and the PCAN-R, combining it to get the PRR plasmid. So it's hard, we didn't talk about that too much because we did the abridged version because we don't have the time and block schedule. It makes it a little bit more difficult. So we don't have time to make the whole recombinant plasmid ourselves, so we start with the recombinant plasmid. Recombinant plasmid, once again, is taking that gene and then putting it into the other plasmid. So I'm going to show you. Here we go. So this is PCAN-R, and on PCAN-R we have that site right here. That's actually the RFP protein and the PBAD section as well. Let me double check. There's the RC, AMP-R, and the ORI site. So on our PRA plasmid, we have the ORI site, we have the RC, remember the RC was the activator, and we have the AMP-R. Now this is our PRA plasmid. In order to make it a recombinant plasmid, we're going to take the RFP and place it into this plasmid. So that's what it means to be recombinant. I struggle with these. Alright, and it's going to go right in between RC and AMP-R. There we go. I should actually have a little pink in front of it. Sorry, you guys saw in class I struggle with these things. Here we go. Okay. Here we go. And almost. Where's Kaylin to do this when you need her? Okay, we're going to pretend because I can't get this one again. So this is our recombinant plasmid that we're actually using. Now we have PRR because we have the RFP, we have the ORI site, we have the activator, we have the so we have the RC and we have the AMP-R. So now this is the recombinant plasmid. That's what we were using, but it was just already given to us, so we didn't have to make our own recombinant plasmid. But what we had to do with this plasmid was we had to put it into the bacteria. So the bacteria that we used was E. coli, and we watched that awesome video from that Instagrammer about why we want to use E. coli. It grows fairly easily, quickly, and it's pretty easy to transform. So that word transformation, gosh, I'm still messing with this because it's going to bug me. Okay, I'm done. The transformation definition was in your pre-lab, so I would definitely read through some of these intros again. But it turns out that live bacteria can incorporate exogenous DNA from outside their own cells into their own cytoplasm. This is how we get plasmids inside bacteria when we make recombinant DNA. This process is called transformation. So a transformed bacteria is a bacteria, or our E. coli, that has our new recombinant plasmid in it. So in order to do this, it was very, very time sensitive and temperature sensitive. You had, let's see, your P plus and your P minus tubes. We got our competent cells. We pipetted our competent cells into both P plus and P minus. From there, we added the protein, the recombinant plasmid, not the protein. We added the recombinant plasmid into our P plus tube, and our P minus tube we did not. From there, we had to do our heat shock, where we put it into the water bath for 45 seconds, and then we iced it after. Gosh, there was a really great image. Actually, you know where it was? It was on that slideshow that I posted. I would go back and look at that again, of what happens to the membrane in heat shock. So that's what we did in order to get our recombinant plasmid inside the E. coli. From there, we plated it, and we covered this today in class. Let me scooch over. So this is what we had as our expected results. So our first one for the LB plate, all we put in was the Luria bras, so we expected high yield of the bacteria for both our P minus and P plus. The reason we expected the P minus to grow was because it's still E. coli, it's just not transformed. But there's nothing about the LB plate that would inhibit growth. So we should have seen high growth in both of those. The next one was our LB amp plate. So now we've introduced ampicillin. Ampicillin is going to kill E. coli that doesn't have the AMP-R gene in it, so it doesn't have the ampicillin resistance gene. So we did not want growth in our P- because we wanted the ampicillin to kill off any non-transformed cells. We did expect less growth in our P plus just because we were going to have it's less optimal conditions and you're only going to keep around your transformed cells. So now we have transformed cells that do contain the AMP-R gene. Now we did notice in class we did have some results that weren't perfectly this, and that's what we discussed why we would have gotten that. Maybe cross-contamination, ampicillin not working is a possibility, but because we got such good results from everyone else, that wasn't what happened here. But the reason we didn't have pink cells here was because we didn't have the activator, which would activate that RFP gene. So in our LB-AMP-ARA plate, that's where we got our awesome results that had our glowing pink bacteria, which were displaying that RFP protein. So those of you that got lots of pink bacteria, what that meant was that you had successfully transformed bacteria by taking that PRR plasmid through heat shock. You put that plasmid into bacteria, and that was what's growing here is what survived and what was actually transformed. So that was our results. They looked pretty good, and that is pretty much it. That's all I can think of. Let me make sure I'm not missing anything. How will you know if you got bacteria, if the bacteria cells got transformed? Well, I mean, there's actually two ways. The first way is if they survived on the ampicillin plate, but that doesn't show if it got the RFP gene. But if it grew pink bacteria, making sure that you understand that word heat shock that I talked about. I had a really good question in class, actually. When did we introduce the ampicillin or the arabinose, which was the ARA? We actually did not. The plates that are already grown with that. So it was already on the plates that I picked up. Okay. I think we are good. Lab 5. So then lab 6 is the protein purification. I'm not going to worry about that one too much because we're only going to do the demo, but you really need to understand the point and what happened in all of labs 2, 4, and 5. I would recommend reading through your intros to all of those labs again, just to make sure you understand it, especially because you have access to the lab manual, so why not? It'll definitely help you out. Let me go to my assessment real quick and make sure I'm not missing any major concepts. But yeah, once again, just remember that I changed the date so that way you can ask me questions next class. Yeah, I think we covered everything. Yeah, we're good. So review those, specifically those 3 labs. Come up with any questions that you can ask me on Wednesday, and you should be good to go. Hope this helped you.